In the name of Allah, hello guys, I'm Muhammad Kushkak with...
I'm Muhammad Jeffrey and we are here to talk about organic molecules. An introduction about carbohydrates and proteins and lipids is most organic molecules found in living systems can be classified in carbohydrates, proteins, fats, and nucleotides. Each of these classes can be identified with simple chemical tests in laboratories.
But the basic structure of carbohydrate is monosaccharide. Monosaccharide is characterized by a group called aldine, and these have a two double bonded, and disaccharide is when two monosaccharide join together.
Lipids include fats, steroids, and phospholipids. Triglycerides are a common form of fats in the world that use the topic of diet with humans. But another thing about triglycerides is about their structure that consists of three fatty acids that attach to one glycerol molecule and temperature and room temperature 25 centigrade. They have some that fats are solid like what that found in animals, but in others that found in plants are liquid. But
we have a protein. Proteins are made up of one or more polypeptides. So polypeptides are made up of one line of actually polymerase. And polymerase are made up of a smaller molecule that's called amino acid. So polypeptides then form that amino acids are joined together with the peptides. And thank you for your attention.
Hello everyone, I'm Muhammad Jeffrey here to talk about mitosis. Let's start the season with some cell cycle process. And now we want to talk about mitosis and its production and preparation of mitosis in different organisms and different animals. Actually, mitosis is the best process to reproduce our tissues that hunters tissues and our cells with mitosis process. Let's continue of this mitosis with my friend Mr. Kushka.
Hello everyone, I'm Kushka and I want to explain for you about the interphase and so M phase. So interphase actually have a three stages: G1, G2, and so S phase. So the G1 actually is occur between the start of the replication of DNA and meiosis. So actually, but we don't have any replication of this replication of DNA. But in a S phase, we have a replication of DNA. Means the DNA of the cells can 2 times more, and so and after that we have a replication, but don't have any change in the number of the chromosomes, and so we have a G2. So the G2 actually is for the protein, so that the protein ready for the synthesis for the division of the mitosis, and after that we have a G0. So the G0 is what? G0, that's the cells that in a G1 don't have any division, and so we can say we have a division of this in a G0.
But the next stage called M phase or mitosis phase. In this phase, we have especially mitosis division in the cell cycle. Actually, this stage will start with prophase. Prophase, metaphase, anaphase, and telophase, 4 main steps of our M phase or mitosis phase in cell cycle. But first one is prophase. In prophase, we have some distribution of chromosomes in the nucleus of the cells. But next step that we want to talk about that is metaphase. The second step of that metaphase called some steps that our cell packet we didn't have cell packet and disintegration of cell packet in this stage. Okay. But next one, we have reproducing of chromosomes, numbers, and cell packet and cell membrane in this stage. But last step with the name of telophase that occurs when we have a complete double cells in this stage, and we have 2 complete cells from more cell or one cell. This is end of cell cycle. But in the last subject, we want to talk about mitosis living tissues. Mitosis in living tissues or onion root tips. We can find mitosis process in onion root tips as far as cool as with what in common garden we can find an onion root tip to study mitosis. Actually, we know mitosis is for developing tissues or reproducing tissues, and these can talk about onion root tips too. Goodbye, everyone. Thank you for your watching.
Bye-bye. Hello guys, I'm Mohammed Kushak,
And I'm Mama Jeffrey
and we want to explain for you
about prokaryotic cells. Okay, for understanding the nature and cell structure and cell function, we should study their organelles, their parts, for their types to know and understand what is the cells and what is nature. To understand the nature, to understand cells, we need to understand ourselves. For understanding cells, for example, we have unicellular and multicellular cells. That's prokaryotic that we want to talk about, that and eukaryotic. But now is about prokaryotic ones.
Okay, so present the bacteria cells are extremely small, approximately 1 until 2 in diameter. Most are heterotrophic and the morphologically bacteria are three types: either row and row shape, and so spiral shape.
Thank you, everybody.